Biology Part 188. Plant responses and the action of plant hormones. Plants are multicellular autotrophic. This is, they make their own food by photosynthesis. This mode of life involves only simple responses to the changes in the environment. Plants do not move actively from place to place and do not possess muscle system. Nevertheless, they can detect and respond appropriately to stimuli including gravity, chemicals, changes in temperature, and contact. The most frequently observed responses are relative growth movement called tropisms. These are directed towards a particular stimulus. Tropisms are controlled by the action plant hormones, as will be explained shortly. Nastic movements are movements of the plants. Parts of the plant are not oriented in a particular direction according to the stimulus. They include certain types of growth movements and a variety of rapid responses brought about by the changes in the water potential in the specialized cells. Our tutorial describes the most important plant responses and considers the interaction of plant hormones in regulating processes including growth, dormancy, germination, leaf fall, and flowering. The tropism. Positive phototropism in shoots. The shoots of a plant normally grow towards light. This is called positive protoprism. As early as 1880, Charles Darwin and his son Francis investigated this response in grass seedlings, which were grown under the different conditions as indicated in our figure. Before interpreting their results, it is important to explain that the grass belongs to the group of flowering plants known as monocotyledons. Consequently, grass seedlings have a rather different structure from that of the broad bean and other dicotyledon. In particular, the first part of the shoot is to emerge from the germinating seed and is a tightly rolled bundle of leaves protected by a sheath called coleoptile. The meristematic cell responsible for producing the new cells required for growth are located not as might be expected near the tip of the coleoptile but near its base, as in the case the leaves of all monocotyledons. At the time of germination, the apical meristem at the tip of the stem is protected deep within the coleoptile close to the seed. What the Darwins discovered was the tip of the coleoptile was part of which was sensitive to the light. Shading the region of the cell division near the base of the coleoptile had no effect, while shading of the tips of the seedlings prevented the phototropic response. Later, investigators concluded that the response must involve cell elongation rather than cell division, and that the cells near the tip produced a chemical substance which caused the elongation of other cells lower down the coleoptile. Some of the experimental evidence for these conclusions is outlined in the figure. When the coleoptile tip was removed, growth stopped. However, if the cut tip is replaced, growth resumed. A small thin sheet of the mica, which was waterproof impermeable, prevented the growth response, but a small block of agar jelly, through which water-soluble substance could diffuse, allowed elongation to continue. In other experiments, it was discovered that a growth-promoting substance could be transferred to a small agar block, which produced curvature of the coleoptile if placed symmetrically at the tip. The growth-promoting substance responsible was the first known plant hormone and was named auxin, from the Greek word auxium, to grow. Auxin was eventually isolated and chemically identified and was found to be indoleacetic acid, The degree of curvature produced in the experiment proved to be an extremely sensitive biological test, and bioassay for the presence of the